All right. Good afternoon, everyone. Today, I want to introduce one of my really good friends, Dr. Rodney Averett, who is an assistant professor at the University of Georgia in the Department of Chemical Materials and Biomedical Engineering. Dr. Averett was educated and trained at Georgia Institute of Technology in the areas of polymer science and mechanical engineering. That's pretty good. And after he graduated from Georgia Tech, he actually took a job in industry, working at Michelin. And from that experience, he learned a lot, as I did, but I worked in industry as a BS scientist, not as a PhD. But nonetheless, he learned something from that experience. And given that, he came back to academia, did a postdoc at Georgia Tech, and then ended up getting a position, faculty position at the University of Georgia. So without further ado, I will let Dr. Averett begin his presentation. Everyone, please mute.
All right, thank you everyone for inviting me. Can I share my screen? Let me see. How do I do that?
You should be all set to share it now.
Okay.
Click on the green. Mm-hmm.
All right. Can you see it? All right, good. All right, thank you for inviting me. It was a reschedule from the spring, and I think that everyone with the COVID situation, it's good that we were able to use this technology to disseminate our ideas and so forth. And I wanted to be able to share with you some of the research that I'm doing on computational and mechanical modeling aspects of fibrin mechanics. When I talk about fibrin, I'm particularly talking about blood clots as they relate to thrombosis and blood clotting. We do some things with hemostasis, or stopping the cessation of blood, and with cessation of blood, but mainly focused on disease states and how clotting is affected. I'll go a little bit into my background. Been an assistant professor, as Juana mentioned, since March 2016. We came up with this sexy name, Chemical Materials and Biomedical Engineering. And that's mainly due to the faculty that we have there. We have some chemical engineers, some material science and engineers, and some biomedical engineers. I've been teaching bioinstrumentation, engineering systems and society. I also have a course now that I'm teaching called multi-scale biomechanics. And that's looking at biomechanics at various length scales, from the molecular scale up to the tissue level or the continuum level. We also do some mesoscale modeling as well. I didn't know the composition of the group today, so I felt that it would be good to go a little bit into my background. So who I am, wanted to talk a little bit about me. I grew up in Columbus, Georgia. Went to Carver High School. As you can know from the title of that, I went to predominantly African-American or Black high school. So one of my favorite things is time travel. So I love time travel and length scales and time scales and mathematics, and I'm just always been fascinated with that. So for my birthday in 2015, I got to dress up as Doc Brown. I'm showing that image here. I like swimming with the dolphins, traveling. I also like doing design competitions and so forth with students. Have 3 daughters, as you can see here. They're a lot older than that now, so this picture is a little bit dated. They're much older than that now. So the little one is actually 7. So this is an older picture. And then I have a beast of a pup that is a toy poodle. He's a vicious dog, so you should be scared of him. And he will bark at you if you are unannounced. So don't mess with him. So Baxter is now 9. He just had a birthday. He's one of my good friends. But I also wanted to talk a little bit about some adversity that I faced along the way. So as I mentioned, I'm fascinated with time travel. So I love time travel. I love Back to the Future. I love anything to do with time travel. So in 1985, the movie Back to the Future came out. And the whole concept of time travel to me deals with XYZ in time and length scales and time scales and looking at the theories, quantum mechanics, and all of these various things. And I'll never forget, I collected a box of trinkets that were made of gears and motors and all types of things when I was in the 3rd grade when the movie came out. And I talked to my teacher about it, and I told her, I said, I want to build a time machine. I said, I think time travel into the past is possible. I think, Doc Brown showed that he could travel from 1985 to 1955. I think it's something that we can do, so I would, instead of playing on the playground, I would go out and take my box of toys out and work with them and try to build, a small miniature time machine. But I was also kind of a class clown too. So she told me, she said, you probably should be a comedian because you're funny. There's no way you'll ever be able to build a time machine. I think you should focus your talents on something that you're good at. You've got this funny dance that you do. I've seen you play football. You're really good in football. And I've seen the way that you can make people laugh. You should be a comedian because you're funny. The funny thing is that, and I'll fast forward a little bit, the teacher, she reached out to me recently via one of her daughters, reached out to me on Facebook and saw that I was a professor now, and she mentioned a bit of this. And she said, well, I remember you had this funny dance and you could make people laugh. So those indelible moments even stuck with her. They stuck with me for certain because I remembered that. And those types of conversations were a little bit discouraging at the time. When you think about a 3rd grader today that wants to travel in time, even though now that I'm a little bit more trained in the mathematical physics and sciences, I don't think time travel in the past is possible. I think it's possible for the future, which is another lecture. But that is sort of demoralizing for a young student to think about time travel, yet to be discouraged by a teacher that is supposed to be leading and training them to pursue higher heights and to pursue higher goals, to tell them that time travel in the past is not possible. So that was the beginning of adversity as I can remember it in the public school system of Columbus, Georgia, that led me to want to pursue higher heights in engineering and sciences. So she told me there's no way you'll be able to build a time machine. And so that stuck with me. As I matriculated through the school system, I'm showing a Google view of my house, the house that I grew up in, 1,000 square feet home. My mom raised 4 kids in a small place in Columbus, Georgia. My mom was a paraprofessional. My dad worked. They worked hard. And I pointed out a statistic here for Columbus, Georgia, to let you know the type of crime that persisted in where I grew up. So I grew up around crime. As you can see, you have a crime rate of about 44 per 1,000, which is one of the highest crime rates in America compared to communities of its size. Columbus is, a lot of people may not know it, but the geographical location puts it within, not many miles of Florida. You can get from Atlanta to Columbus pretty quickly. You can travel from Alabama to Columbus. So there are many ways in and out of Columbus that make it attractive for the drug trade and a lot of other organized crime that you see in larger cities. But it's just that where it's located and the size, a lot of people don't know about it. But I grew up around crime. The pecan trees that you see in this photo are pecan trees that bloomed every fall season, even around now. you can't see it, but the street that goes here is Ewart Avenue. And what I would see, you can't see it in this photo because they're not showing it, but there were drug dealers and there were people who walked up and down the street all the time. And it was so bad that the people who used drugs would steal the pecans out of our own yard and try to sell them back to us. So these pecans would fall in our yard and the people would try to sell us our own pecans back. So, I mean, these are stories that I remember. And, you know, I went to school not far from here. So it was less than a mile from here that I grew up, and went to Marshall Junior High School. And then I went to Carver. And as I went through school, I eventually, as I pointed out here, I was the valedictorian of my high school class. I had many offers for scholarships, Iowa State, Georgia Tech. I mean, I had to work hard to overcome a lot of those challenges. I ended up attending Georgia Tech for my bachelor's, majoring in mechanical engineering, and pursued a bachelor's in mechanical engineering. And then going along the way, as Juana mentioned, I went into industry. So when I decided to go into industry, I wanted to first pursue automotive industry because I talked about Back to the Future and cars and all of that. I thought it would be a cool thing to do research on cars. I mean, I thought it was great, right? I mean, you would be able to design your own cars. I can work on drawings. I can do all of this cool stuff. So they gave me a project to work on the new Acura TL. That was a part of the interview process. I was like, this is going to be great. I never forget, my manager took me into the room and he started asking me all these questions about beam deflection and what is the equation for beam deflection for a cantilever and all. And I'm like, okay, yeah, I'm doing the work. But I'm like, okay, cool. And we found out, because we had an HR manager that was there in the room, and she said, you know, what he did to you was just racism. He's never done that to anyone. He's never asked anyone about beam deflection. He's never asked them about the equations of beam deflection. He's never asked them about the bending moment. He's never asked them any of these things. The fact that he did it to you was just racism. And I remember her name. I won't say it here because you are recording. That's why I said it. But anyway, I mean, but she basically got on to him. She was a part of HR, and she later told me, she said, you know, I just wanted to let him know that what he did was not cool. He's always asked very simple questions, but for you, he did, you know, of course I got the job and I took the job, but the same guy, I went and I told him, I said, I'll call him Larry. I'll just make up a name because I don't want to say his name. I said, Larry, you know, I see that there's a potential to go on to graduate school here to pursue a master's degree in engineering. I said, I want to take advantage of that because if the company pays for it, I would like to go to Ohio State. I was in Ohio at the time. I can tell that part. Maybe they can put two and two together and figure out who this is. But I think, I think so. I said, I want to go and take advantage of this. He said, well, you know, I don't think you don't go to graduate school and get a master's degree. It won't help to advance your career. I said, Dave, now don't you have a master's? Well, I called him the wrong name. It's supposed to be Larry. I said, Larry, I said, don't you have a master's degree? He said, well, yeah, I do. He said, but you shouldn't waste your time going to get a master's degree because it won't help advance your career. I said, So, you know, I pursued the applications and so forth anyway, and looked at the best program for me and looked at some of the potential opportunities at other schools or whatever. Ended up deciding that I wouldn't take Larry's advice, and I decided to go back to graduate school and to pursue my degree. So after going back to graduate school, I said, Larry, forget you. I want to learn about mechanics, and I want to take what I do and learn about mechanics, which is something that I started out learning about. So I started out very simple, looking at micromechanics at the fiber level and looking at fraction mechanics. So I pursued a master's in mechanical engineering with mechanics and materials focus. with a minor in mathematics. And one of the first things that I looked at was fatigue. So instead of looking at how materials behave under quasi-static loading, I was interested in learning how they behave under repeated loading. So I'm saying not from a quasi-static standpoint, but we were interested at the time in the large rotorcraft and airplane division, and saying, okay, how can we reduce the weight of airplanes, airplane tires? And so in order to do that, we had to understand the mechanics of how these composites behave. So that's one of the first projects that I undertook in graduate school. I was allowed to pursue this, learn about scanning electron microscopy and all of the tools that you need to evaluate the materials. Ended up, was able to publish some papers in this area. Looked at linear elastic fracture mechanics, critical flaw size. So we're building a foundation now of how do you structurally evaluate materials and composites and look at the energy required to break the structures at the micron length scale and look at the critical flaw size that leads to failure. Was able to publish another paper looking at now nanocomposite fibers. So now I'm basically interdispersing nano materials within these fibers. So we looked at spherical nanoparticles, we looked at vapor-grown carbon nanofibers, we looked at carbon nanotubes, we looked at multi-wall nanotubes, we looked at single-wall nanotubes, all of the various things. And we came up with a structural validation technique that allowed us to evaluate what the maximum stress is that leads to fiber after the materials have been cyclically loaded. So now I'm looking at an experimental technique that's able to assess what the residual strain is in the materials. We're looking at scanning electron microscopy. We're looking at wide angle X-ray diffraction. So all of these techniques that are used to evaluate the materials. And so here again, looking at this, I was able to determine that the under fatigue loading, this is not quasi-static loading, but under fatigue loading, the structures, they basically exhibit defibrillated surfaces and they have fracture that is not characteristic of normal quasi-static loading, which was something that hadn't been published at the time. But it was just basically due to the fact that the carbon nanotubes or the vapor-grown carbon nanofibers in them, they basically act like flaws under fatigue loading. So they basically act like a defect in the case of fatigue loading. So that was something that I was able to confirm even with TEM. When you look at the fiber axis and the alignment of the fracture initiation angle in the fiber is reminiscent of the nanofiber orientation angle. So it's basically that image that you saw is basically failing in the same angle as the dispersion angle up in the material. So I said, okay, that's all cool. And Juana talked about me taking this to industry. So I sold this idea to a company that I did go to Michelin. And you know, I jumped right in head first. I thought that the ideas that I learned could be applied not only to materials, but I thought they could be applied to composites, which a tire is a composite. So I talked about starting with tires, and we're looking at mechanics. We're looking at we're looking at fiber mechanics, looking at mechanics of structures. We're trying to basically reduce the weight of materials. That's the overall goal. That's why we were using a lot of the carbon nanotubes, the vapor-grown carbon nanofibers, and the multi-wall nanotubes. So by the end of my dissertation, I started looking at artificial neural networks and how you basically could use this model or these models to predict when the materials would fail based upon a state variable-based technique. So instead of looking at the history base of the particular material, I was interested in looking at the state of the material by looking at some type of state variable. I was interested in saying, okay, when would the materials fail? So this led to some work to where I was basically looking at certain input variables to where I can predict some macroscopic output variable. Michelin was very interested in this because they had not only some materials that they were looking at, but they were also looking at monitors for various tires and other components in the tire manufacturing industry. So this went over well. I jumped right in head first. I was able to apply this to some viscoplastic modeling that they had for large tires. I was able to, and the reason why I joined is because it was a long-term project that was allowing me to travel. I was able to go to the UK. I was able to go to Hungary. I, went to Eastern Europe for the first time and spent a lot of time in Hungary and Budapest, and so that was great. And that's basically why I took it, is because it was supposed to be a 6-year-long project that was allowing me to use a lot of the tools from my PhD to help them advance their large tire line to increase profits for the large tire division. And these tires are very profitable because they're so big. I mean, because of the amount of material that is involved, they have a large profit margin for the company. So I was able to use this using artificial neural networks to look at elastic modulus, how it degrades as a function of residual strain, looking at the maximum fatigue stress for both unreinforced and reinforced materials. So that was published. We were able to basically publish that once I got there. And like I told you, we were going from basically a history-based approach to a state variable-based approach. And then I was able to use some genetic neural network algorithms to predict what the mechanical behavior would be for structures that have a certain amount of fatigue stress based on this state variable. So that was the conclusion of the research that came from that. And that's very important, right? Because if I can predict the constitutive-based response of the material subsequent to some type of damage accumulation, that is very important to manufacturers who are trying to develop the next state or the next best material. So the next composite. If I know what variable to look for to test for from a macroscopic sense, I can basically use that to tune, to basically be able to say, okay, structurally, how will this material behave once it has been subjected to these fatigue loading conditions or this loading condition. And the tire is a perfect model because it undergoes fatigue all of the time when it goes through in and out of a contact patch. So it's basically fatiguing. As you're driving down the road, it's fatiguing. So it was a perfect model for testing a lot of the work that came from my PhD. So basically using these models, I was able to predict the mechanical behavior, using genetic artificial neural networks. So this was great. And everything was great. I mean, I learned a lot. A lot of my work was being used. I was able to predict the mechanical response, and using the genetic neural network algorithms and predict that and match it to the experimental response. So while in the industry, I never forget, it was it was after Halloween. So we're going into Thanksgiving. We're going into Thanksgiving, and Juana brings up a very good point in her introduction about going in the industry. And I'll never forget, the highest officer of the land at this time was being pursued by an African American. And I'm working in Greenville, South Carolina, so I won't say any more about that. But geographically, you know where you are, and I'll declassify stuff. I don't care. Let them watch it. Maybe I'll friend them on LinkedIn after this. But what I will say is, I think the industry has to know, so I like I said, I took the job, and I thought that racism, the barriers of racism, you start to believe that certain things will topple or crumble once you get a certain amount of letters behind your name. And you start to think that when you walk in the door, there's instant validation and instant credibility and sustain credibility that you don't have to validate yourself on a daily basis. You don't have to walk in the door. If I have the degree, I don't have to go in every day and say, hey, I got the degree again. I get a test every day I walk in the door. You know, so glad. And it was. Now we're in Thanksgiving. The election is going by now, so Barack has already won. People were quiet. And I'll never forget, I wanted to go to one of my, I had some Thanksgiving stuff that I wanted to do a little bit earlier. And my boss, he said, I said, well, you know, my daughter's playing a role here in this play, and I want to go ahead and try to leave out a little bit early. I never left early, but I wanted to leave early for this date. I said, she's going to be in a play, and she's got to be, you know, a pilgrim in this play. He said, you know, I don't think there were any black pilgrims. He said, he said, so why are you going? He said, I don't think there were any black pilgrims. I said, I call him Larry. I said, Larry, now, what do you mean by that? He said, I don't think there were any black pilgrims. Then he tried to laugh it off. I never let that slide because I basically did mention it to some people, but that stuck with me because it allowed me, and you know, he made some other comments, which I won't reveal all of that, but I let them know in a nice way going out. But there weren't any black pilgrims, so you begin to see that this racism is pervasive, and it is rooted deeply and ingrained within the corporate American culture that you feel, you know, once you get to a certain status that maybe you won't have to deal with things like that. So that was a defining moment for me once I got back in the industry, because the Larry that I knew at Honda was the same Larry that's at Michelin. It's just years later, you see? And I'm quite sure Larry is still in 2020 in a lot of these places. It's just the fact that society just hasn't dealt with Larry. So I'll say that as a lot of you, and I'm not discouraging the industry, because it happens everywhere, but what I will say is, you know, pursue your dreams, but there will be Larrys along the way that will try to test you. They will try to throw you off your trajectory. They will try to make you feel that you are an imposter. They'll have you leaving trying to figure out if you're an imposter with the imposter syndrome, starting to think, well, maybe I'm not good enough. Maybe this job is not for me or whatever like that. So I say, you know what? Here again, I'm going to learn all I can. I say, forget Larry. I'm going back to learn about biomedical engineering. So I said, you know, I'm not, forget Larry. You know, I'm not going to worry about him right now. I'm going to go ahead and take a lot of the work that I've learned about fiber and fibrin mechanics, and I want to do something great because, you know, my mom and my grandmother would always ask, well, what do? You know, you just sell things that are black and round. You went and got a PhD. You selling tires, baby, you know? And so, you know, I said, no, I'm actually doing research that's cutting edge, you know? And so they said, well, you know, I thought you were a doctor. I thought you, so, well, you know, biomedical might be a great fit for me. So I said, you know, maybe, you know, so, and I can tell this story, you know, Juana has been a great colleague of mine, but I never forget we were at MIT. That's another story, because there were some people up there like that too. But we were at MIT, and I'll never forget we were on the same page. One is very ambitious, you know, like I am, and we just gravitated to one another. And she's, you know, really, really adept in polymers and so forth. And so our backgrounds crossed, and she was telling me about some stuff she was trying to do in biomedical. That's a longer story than we got time to talk about. But anyway, I was introduced to a lot of the biomedical field because of her. And, you know, we got to talking about, well, maybe you should try this and think about Emory and think about this and that and so forth like that. So I said, that sounds like a good idea. I didn't know anything. I barely knew what a cell was. I mean, a prokaryotic versus a eukaryotic cell. I didn't know. I barely knew. So I said, well, I'm gonna have to do some work on this. So I said, well, what's the best way to do some work? It's like, if you want to be a coach, right? Maybe I need to go and coach some high school students. So people don't know this story. So I said, you know, I'm gonna have to take like 6 weeks to go teach like a biology class. I did that in a high school before I decided to take a postdoc, for real, because I didn't know. I mean, I barely knew how a virus behaved. You know, I didn't know anything about a mutation. You know, ATGC was just ATG and C. You know, so 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 I said, okay, you know, I'm gonna try this out. So I basically went and decided to pursue a postdoc. And one of the projects that I was looking at was blood clots. I said, well, clots have to be similar to what I've looked at because they're fibers. I mean, they've got fibers. They're just bio systems, right? Instead of them being synthetically made, cells are making them via some extracellular matrix composition. So there has to be some overlap. So I said, well, yeah, this may sound like a great idea. You know, I can start to look at how blood clots develop. Blood clots are associated with various disease states and all this stuff. So that sounds a lot more cool. Maybe my grandmother and my mom will listen to me now if I talk about what I'm doing. And maybe they won't laugh at what I'm doing. So I said, okay. I basically decided, okay, I'm gonna do a postdoc with GT and Emily. And instead of looking at it just from a biological standpoint, I said, well, I'm going to apply the micromechanics that I'm using to these systems and the modeling that I'm using to these systems. So that led basically to doing a postdoc and looking at clot structures as a multi-scale structure. So instead of saying now I'm looking at it from a holistic or a continuum level composite structure, I have to start now because of the protein structure that's involved. I'm having now to look at them from the single protein level up to the macroscopic scale. So I said, okay, that sounds good. Was able to get some funding for that. And so the hub of my research now is focused on computational, being the large component of my research, imaging and experimental. The experimental and the imaging are only there because people sometimes don't want to collaborate. So I'm stuck not having experimental results. So sometimes we have to do our own work for validation. But in reality, I don't really like doing it. But that's why I have people like Juana to collaborate with. She loves to do that. And I like to do computational stuff. So it works out good. And she answers the phone. So that's what's good. So everybody doesn't answer the phone, right? Or answer the emails. So, but I've decided to take a computational aim and a computational focus on my research. So I have 3 things that I basically look at for the computational and mathematical modeling. So we talked about neural networks. I said we're going to combine some mathematical techniques with the neural networks and look at how computational and mathematical modeling play in with the single protein level. So at the single protein level, I use what's called the computational microscope. And I worked with some guys at the University of Illinois Urbana-Champaign to basically be able to perturb single proteins without using an AFM. The results correlate with what's perturbed by a single AFM system, but it's just the fact that I'm using a computer and I can perturb small molecules, small proteins, and we can even probe things like 20, 30 amino acid residues long. So I can and that's something that's hard to do even in the lab. It would be very hard to do that. So I'm able to look at those structures. If there are any type of mutations in a gene that causes an alteration in a protein structure, I'm able to look at those. I can perturb those structures and say, okay, mechanically, how do they behave differently than another material? So basically using a computational microscope to do that. I'm also using imaging analysis at the macroscopic scale to, and I don't know, this video might not play, but I also look at the macroscopic level, things like lysis. So how fast the structure is lysing or chemically degrading as a function of time, as a function of enzyme concentration, looking at various things like that as well. So the main focus of my work is on two sides. I'm looking at both hemostasis and thrombosis. Of course, hemostasis, I want to stop bleeding. You cut yourself, you have a cut to the endothelial lining. Basically, it initiates coagulation cascades, and you have 3 steps there. You got vasoconstriction, platelets go to the surface, and then you form a blood clot. I'm interested in the 3rd step, in which you basically are looking at the structure and the dynamics of blood clot formation. Coagulation cascade is very, very, it's complicated. And here again, I'm looking at this final formation of the fibrin clot. But the venous thrombosis side is something that affects a lot of people. So I'm basically looking at how venous thrombosis affects various patients based on their age. So some of the things, the 2 diseases mainly that I've been focusing on are sickle cell disease and diabetes. And some of them have a genetic component, but mainly they may be multifactorial in nature. So they may have to do with age or surgery, malignancy or whatever. But in the case of sickle cell, of course, that's a genetic homozygous SS condition that you have to have 2 parents or whatever. So that's a genetic condition that we're looking at blood clot structure and how the clots are alterated in sickle cell disease patients. I won't go through a lot of this, but this is just talking about clinical treatments, and we want to basically be able to advise pharmaceutical companies on what and how they can develop more and better pharmaceutical treatments based on the ones that are currently in the clinics right now that they're using. And so one of the questions that I really want to ask is, how is clot structure related to disease? All clot structures are not the same. You have a clot structure from a patient with sickle cell disease versus having a clot structure from a patient with venous thrombosis versus having a clot structure from a patient with diabetes. They're all different. They behave different mechanically, and the origin of the mechanics and the clot rupture and the dislodging all emanates from the molecular scale, which is why I'm so interested in the molecular mechanics of clot structures. So from a modeling standpoint, it would take too long for me to be able to do a lot of these experiments in the lab. So I'm interested in, at the molecular scale, this fibrinogen structure, which is the main protein. It is a 340 kilodalton protein that is about 46 nanometers in size. It's a hexameric protein, so it has 2 sets of trimeric, so it's a double trimer basically that's connected via a central nodule. And it is very rigid, so it strain hardens under tension, and the enzyme that activates it to start the coagulation cascade is shown in the middle here, which is thrombin, that causes a collection of proteins that assemble in a half staggered assembly to form a blood clot. So we basically have, that's what I'm interested in, but also in the clot are red blood cells, which I'm showing here, which, as you know, red blood cells are composed of hemoglobin. So I'm also interested in how hemoglobin behaves at the molecular scale, because of course, sickle cell disease is a disease that affects the red blood cells. And when the red blood cells get trapped within the fibrin mesh, it causes the mechanical behavior of the structures to behave differently. Hence, I'm interested in looking at the nanometer scale to the micron scale to the millimeter scale. So I'll talk a little bit about some of the things that I've done computationally. So here we're down at, of course, the nanometer scale. And I'm looking from here all the way up to the micron and even beyond the micron length scale with the research that I'm doing. Okay. All right. So length scales are important for blood clots. That's one of the main topics that we have to drive home. That's where my whole Doc Brown analogy comes in about length scales and time scales, and how length scales and time scales are important for understanding how materials behave from a multi-scale perspective. The spatial scales that we're talking about go even beyond, like I said, when you just look at a clot, but you have to think about the molecular unfolding that occurs at the nanometer scale, and to say, okay, how does that molecular unfolding contribute to the clot extension? How do those helical coil regions, how do they dictate the mechanical behavior of the clot at the molecular scale? And to add on top of that, that is for a normal patient. What if you have a patient that has a disease? So then you would have mutations, or you may have alterations in protein structure. How does that clot extension behave? Is it more rigid? Is it softer? Will the person bleed to death, or will they develop a heart attack or a stroke or a pulmonary embolism as a result of the clot structure being different? And those are the questions that I want to answer. So one of the first things that I was interested in was looking at how a single domain on the protein structure governs the stress-strain behavior of the fibrin mechanics. And this was a great project because it allowed me to look at the molecular dynamic simulations and the computational microscope and use as a validation tool an atomic force microscope to validate the mechanical behavior. So basically, I took this model that is it's actually a three-component model, because it's using steered molecular dynamic simulations and the computational microscope, but it's also using polymer physics and looking at polymer physics and how you can use polymer physics to figure out what the strain is in a system based upon the chain orientation and all the polymer physics associated with fibrinogen and fibrin. So I was able to do that, able to validate it. It's not shown here, but it's in the paper. I jumped off track, but I'm trying to cut this down. Anyway, I said, okay, let's go ahead and now take this to another level. In order to develop this multi-scale model, if you go back in time, looking at the 70s, you have what was called rate kinetics and convection diffusion reaction equations. Those equations were great mathematically, but they really didn't take into consideration a lot of the structural analysis and a lot of the protein behavior analysis. You have a lot of differential equations, you have a lot of the governing equations that look at convection, diffusion, and reaction, but they don't necessarily take into consideration a lot of the structural information. Of course, Navier-Stokes and computational fluid dynamics allow us to look at fluid flow and if blood is flowing, and looking at the transition from laminar to turbulent, all of these various things. But as you go and get into the 2000s with the advent and with the advancement of computational power and computational capability, we're able to do things like dissipative particle dynamics, DPD simulations. And we've done some of this, and I'll show a little bit of it. Cellular POTS model, and here, like from 2010s up until now, multi-scale models that allow me to take that single protein structure into consideration and how it governs the overall macroscopic behavior of the material. So because I have access now to faster computers, more nodes in the cluster, more computers, more time that I can, have to predict these things, you can do all atom simulations that will allow me to predict that behavior. So you look at the literature, going back now, fibrin formation is now starting to be studied from various researchers. Still not a lot of people doing it computationally. A lot of people are still focused on the experimental aspects. They're looking at fluid particles, red blood cell particles. And one of the things that, here again, that I'm interested in is this all atom simulation that I talked about, the iterative Boltzmann method, and looking at a coarse grain model and fitting that into a multi-scale model with the reaction equations. I never said that they weren't important. I just said that they weren't enough to be able to govern the entire behavior of the system. So if you combine the coarse grain system that has the all atom information with the iterative Boltzmann method, and you combine that with the governing differential equations, you can develop a true multi-scale model that's able to capture the mechanical behavior and the mathematical physical behavior of the material, which allows me to predict the final fiber clot structure. So if we go a little bit further, that's exactly what we've done now. Taking the glycoprotein, 46 nanometers, and basically this is some of the work, this is some of the research from one of our publications. We're able to predict how this assembles. So from a polymerization standpoint, I'm able to predict this double-stranded fibrin oligomer structure. The long double-stranded protofibril lateral aggregation is something that I'm able to computationally assemble and to mimic in basically just like it would be in an in vitro system. Basically using a combination of DPD mechanics that I talked about, I can take into consideration the molecular weight of the single proteins. And this is one of my coarse graining procedures that I'm using. I'm starting with the all atom model and basically running the iterative Boltzmann inversion, all of this. We can talk about this later, but, and a lot of this is probably more than I have time to go into, but this is just basically one of the ways that we're basically able to predict this final structure using the final coarse grain model, taking into consideration the bond angles and all of the bond distances, all the equilibrium distances from the intramolecular parameters. Compare that to what's in the literature. I got a lot of slides, so I'm trying to start flying through that. So I see it's 452. And then I'm continuing to go and for the multi-scale model. So here's my reaction kinetics modeling that I talked about. So now I'm taking into consideration my reaction and kinetics modeling. We have a lot of literature from that goes back into the 70s and the 80s that people talk about. Here's this differential equation that can say, here's your tissue factor and how the kinetics behave with this. Here's a differential equation for this. Here's how factor 13 behaves. Here's how factor 3, how factor whatever behaves. So we've got these governing differential equations, but no one has really put them into a model that encompasses material structure as well. So we take all these reaction kinetics equations. So here are my differential equations that govern the mathematical behavior as a function of time. So I'm interested in looking at how these different factors and enzymes, how they behave as a function of time. So I have all of my reactions, kinetics equations. I know, and what's on the left are some of my parameters that govern those reaction constants or reaction parameters, and some of the parameter values. But if you have a disease, of course, some of these parameters are going to be different. So we have to take that from either the literature or we have patient data that we can take and use in place of what's here for normal patients. So that's how we basically turn it into a realistic model that's not just a mathematical model, but it does have some type of experimental or clinical relevance. And also here, if you look at the parameters, I'm also working with canines right now with canine diabetics. So some of these parameters can be taken from canine diabetics. We look at our reaction kinetics modeling, and then basically we're still trying to get the multi-scale model. This is my polymerization that I also can mimic this knob-hole interaction for when the fibrin clots form. And this is just too much, but another thing I'm able to do is when you think about the enzyme or the catalyst that causes the systems to polymerize, I'm also able to look at how the enzyme controls the reaction rate and the polymerization rate computationally, which is something that hadn't been done in the literature as well. So thrombin is the actual activating enzyme that causes the coagulation cascade to initiate and causes the clots to form. So of course, higher thrombin concentration is going to cause faster reactions. Lower concentration causes the clots to form more slowly. Higher thrombin concentration is more present in sickle cell disease, diabetes. So we're basically interested in looking at not only structure, but the reaction rates and the polymerization rates of how fast the structures form. So this is looking at the simulation times going through time of how the clots form with high thrombin concentration, low thrombin concentration, and how the branches form, how the fibers form, how this is looking at a 400 nanometer box. So we're zooming into this box and we're looking at structural entities of how the fibers form and how the branching occurs with the fibers. And we even go out to 1,000 nanoseconds. And by the end, we're able to basically look at and compare what the structure looks like, the structural characteristics of what the structure looks like, and we compare it to what you see in the literature. And we see this 23 nanometer half staggered assembly that I talked about earlier that forms. So computationally, we're able to structurally represent what the material behaves like. And this doesn't take as long to do as what you would do in the lab, but we see the bilateral junctions, we see the 14 fibrins per strand. And so from a physiological standpoint, I'm able to mimic what you would see physiologically using a computational model, taking into consideration all of what I talked about. I've also done some work with some researchers to look at the elastic modulus. Of course, I told you about the elastic modulus that I worked on with Martin at Wake Forest. The elastic modulus is in sync with what you would see in the literature, and the fiber lengths. Everything is about what you would see computationally as what you would see for experimentally. I'll go through this. Dissipated particle dynamics is good because I can basically cut my computational time down. I can use a different bead diameter. I can cut my simulation time down and represent the system. Let's change a little bit up to hemoglobin. I talked to you all about sickle cell disease. I'm interested in hemoglobin characteristics. Of course, if you have a sickle hemoglobin molecule in comparison to a normal adult hemoglobin molecule, I'm interested in the compression characteristics of this, because when the red blood cells sickle, they turn into a crescent shape and they become more rigid and they have to squeeze through the vasculature. So I'm interested in the mechanics of that. So I developed the hemoglobin compression method for looking at how hemoglobin molecules behave at the molecular scale. That's looking at sickle hemoglobin. It's looking at normal adult hemoglobin. It's looking at deoxygenated hemoglobin, oxygenated hemoglobin, and glycated hemoglobin for patients with diabetes. So I don't have all of those results, but so, well, actually I do. So here's deoxyhemoglobin, oxyhemoglobin, carboxyhemoglobin, and glycated hemoglobin. As you can see, the stiffness of glycated hemoglobin. So if you think about someone who has diabetes, even with controlled insulin, their stiffness of their hemoglobin and the resulting morphological characteristics of their red blood cells are going to be more rigid. And that's shown here in red. You basically see that this is showing the anisotropic characteristics of the materials in the X, Y, and Z direction. So I'm compressing them in different directions. And I'm looking at the mechanical behavior characteristics of how they behave under compression. So as they squeeze through the vasculature, the materials have a higher stiffness, the lowest being the carboxy, and then the next stiffest in most cases was the oxygenated. So that was very important because this basically just shows that for someone who has diabetes, like I said, even with insulin control, you have higher red blood cell stiffness. So here in the conclusion for this, I've developed a fibrin clot model using reaction kinetics and some other mathematical techniques. Use a reactive dissipated particle dynamics model for simulating fibrin clot mechanics, looking at the effect of also low and high concentration of the enzyme thrombin that catalyzes and cleaves the fibrinopeptides on fibrinogen. Future direction is to combine some of the other things that I talked about, like Navier-Stokes, to look at other things and look at wall clot interactions. Attachment to endothelial cells is something I'm trying to get into, and how they attach to endothelial cells, because they basically have to attach to something and break off from something, hence causing something like a pulmonary embolism or a stroke or a heart attack. And that's so other things I'm looking at is looking at single fiber mechanics. And I've got some cool videos here that we're basically doing. We're looking at how you know, basically a lot of these simulations take a long time to do. So I kind of feel proud to show this. It takes like 3 weeks. So it's like all random stuff that I'm combining. That's what we get to show. We don't get to show like other stuff. So we can show stuff like that, right? But you know, but as modelers, we have to show something too, right? So other things like hemoglobin compression. I talked to you all about sickle hemoglobin. This is sickle hemoglobin. So if you have sickle hemoglobin, this is what it would look like under compression. Isn't that cool? It took like 3 weeks. For real, I mean, you know, it takes a while. But this is because this is like 6 different hemoglobin molecules. And basically, these are 6 HDS molecules that I'm using. And actually, the editor made me do that too. Because he wanted to know how not just one molecule behave, but how multiple molecules behave. So those are some of the cool things we're working on. Looking at long scale simulation times using reactive dissipative particle dynamics, and looking at that final structure that I talked about. So all of those simulations, they end up in a final structure. They take a long time to get the program right, but once the program is developed and it's running, we can do a lot of cool things. But I want to challenge you all now to become Nobel laureates in physics, physiology, medicine, and chemistry. Don't let Larry deter you from what you want to do. Don't let Larry say to you that you can't become a Nobel laureate in physiology or medicine or physics or chemistry. They had an article last year about why are there no Black Nobel laureates in science. I challenge you that today there will be, coming out of the great institution of Morehouse right now. Somebody will hopefully hear this and hear my story about how Larry treated me and say, you know what, I'm not going to take that. I'm not going to let Larry continue to talk to me this way. I'm going to go in the lab and develop something new, something exciting, and something intriguing to get the Nobel panelists to select me for a Nobel Prize. And we've had some Nobel prizes, of course, in peace, and that's great. We need some Nobel scientists as well. And so that's my challenge to the young academics today that are pursuing research careers and careers in science and technology. I want to thank some of my collaborators, Dr. Wana, XQ, one of my colleagues at UGA, one of my postdocs now who is a faculty member, the GAC-RAC, which I am tapping into all times of the night, which is my supercluster at UGA, and then of course the NIH. So thank you for having me for this presentation, and I hope you all learned something. I don't know. Thank you.
Thank you, Dr. Everett. Really appreciate you.
Phenomenal.
Say again?
I said that was phenomenal. That was,
okay.
It was dope. It was real dope.
Yeah, it was very interesting.
So we thank you for bringing it all home. First, Dr. Lawrence looks really scary.
I was
right
just
now.
about to say, Brian.
That's why I turned off my video. So I'm going to turn it back on.
Dog, like for real, Brian, for real.
Like a little Jason-ish a little bit, but
oh no, that's so evil.
Thank you, Dr. Everett. That was great. You know my brain was stirring. So I got like so many ideas coming from that, but more importantly, I think we're going to open the floor for questions here in a second. But I hope it hit home with the students that your path, similar to mine, how you started out working on something completely different, right, and kind of did a 380 to get into a whole new area, right, and that says a lot, and you did the work, right? So you did the work. So students, I will open the floor. Do any of you have questions before I'll let you go first before we ask any?
Okay, look like they're silent. I do have a question. I have one. I'm jumping at.
Okay.
I have a question. Do you ever measure tensile strength
We
of
do. Go ahead.
the fibers, and how do you do that in a computational? Uh, like, how could you? Yeah, how do you measure that?
So I did mention I have a colleague at Wake Forest. So experimentally, let me answer your question in two different ways.
Yeah.
I have a colleague at Wake Forest that has two customized AFMs that we basically polymerize a clot on a substrate. And I can show you a picture. I just didn't put it in here, but I have a picture of basically you polymerize it on a substrate and you take the AFM tip and you basically just pull it. So I can get all of the mechanical properties like that. That's what I use to validate the model that I talked about when I did the mechanical model. So starting with the molecular structure, I scaled that up to a fiber model and then matched the character. I just don't have a slide in here, but I can send you the publication after this, and you can see the computational that's matched with the experimental results. So I was able to take that single fiber behavior and then using the computational microscope, I was able to elongate it like that. And then separately, I did the experimental. So it takes a little bit of fidelity to do it because when you polymerize it on the substrate, the fibers have to be orthogonal to the channels, and it's like an art to it. It's kind of tedious because you got to basically get a microscope, you got to look down on it, and you got to take the AFM tip, it's got to line up, so yeah, you can do it, but basically what Martin had to do, and he does single fiber mechanics, and so he's my guy that I go to for if I need to do any of that work. He basically has the two, but he had to custom. It's like getting a car and customizing it to, putting your own engine and transmission and stuff in it. You know what I mean? He had to basically take it and customize it for that.
So he had to engineer the system.
Yeah, he had to engineer one that he bought. Yeah, and he had two of those. So that's how he's able, and he's kind of, one of the only people that does that for single fiber mechanics that are biological fibers. Yeah. That are, ECM fibers. I mean, he's done collagen stuff and all types of stuff, but it's single fiber mechanics looking at tensile strength. He can look at the modulus. He can look at the strain hardening behavior. All of that, comes out of it. But
I mentor students for science fair projects, so I'm a science fair coach as well, and I have just looking at the tensile strength of synthetic hair, canacolon hair.
Okay.
Just this presentation was kind of right on time because I saw some other things that you can look at and kind of talk about. And because they're doing it virtually, virtual science fair, I'm just trying to think of other ways in which she can show her interest and show her intellect. So that was just interesting. So that's why I asked you about tensile strength. And another thing, I just want to commend you on your journey. I spoke with my students this morning in analytical chemistry about the trajectory of success, and that as long as you're moving forward, don't worry about the low points, because there are some times where you have to, change direction or things don't go your way. Life happens.
That's true.
You go down, you speed up. You have children. I have
That's
5
true.
sons, so life changes when kids are involved too. And but I thank you for confirming what I literally just spoke this morning at 11 o'clock to them, and that's that your trajectory to success is your own, and own it and be proud of it regardless of the failures. See them as lessons and continue to move forward, and that everyone's success doesn't look the same, and the people who you think had this like straight trajectory never do. You know what I mean? It's always some hills and valleys. But I am grateful that Juana actually recorded this, because I have a son that's an electrical engineer and he's looking into doing a career kind of change. And it's like I could have kicked myself that I didn't even send him the Zoom link, because he needed to hear you and your pathway through, because he has been kind of uncertain about where he wants to go, and that he's totally changing. He's doing UI UX design now.
Okay.
Right? So he's into user experience and all that kind of stuff, and he's a creative, but he's also an engineer, right? So
sure.
I'm really begging you if you could just stick this.
No, I got you. I'm definitely. It's actually opened up. This whole dialogue is opening up some stuff that I think the timing. You were supposed to present in the spring, but it's emotional for me because it's like it's perfect for right now. It's perfect for right now with what's happened over the few months and just you telling the story. And then, so I'm a coin this statement from you to say that Larry is the new Karen, right? Because Larry is the new Karen. That's what I learned. But students, don't leave me yet because I'm going to assign a poll for you. But Dr. Derricott, you can go ahead and ask your question. I'm signing the poll, students, while we're doing a little Q&A. You can complete the poll, students, please.
Gotcha. Okay, so Dr. Averett, thank you for the presentation. This was excellent. I definitely, co-sign the sentiments from my colleagues as well about, your message to the students and whatnot. I had a few questions for you. So the first one that I had was, you showed us a few of the details of your artificial neural network, and you showed us that there was like an input vector that goes into it. And I know with all of these algorithms, there's always, a myriad of different inputs that go in to train these algorithms and get them to perform better. But can you give us a sense of what type of inputs go into that input vector? Sure. I know it could be a lot, but just like give us a sense of at least a few.
I want to go back because I want to basically look at one in particular that you all can basically, so let me, I don't know. Can you still see my screen?
Mm-hmm. Yeah, we can still see.
Okay. In particular,
you should be able to share it. Yeah, you should.
Is it shared?
Oh no.
Yeah, it's already sharing, but
oh, I don't see it. Oh, there it is. It's me. I think I'm doing something wrong. So yeah, it's there. Sorry.
In the case of this one, this is one that I was interested in looking at, leading up to a lot of the tire dynamics and mechanics that I was looking at. So in this case, my input vector space is going to be the ratio of the minimum and the maximum stress. So the minimum and the maximum stress that under fatigue loaded, and then the amplitude stress. So I can have a minimum and a maximum, but then the next variable is my maximum stress. And then from the actual material characteristics in this particular example, I'm looking at the underformed modulus. And then this state variable, which is very important, which is a mathematical entity, is a residual strain. So all materials under loading, once the material irreversibly deforms, you have what's called a residual strain that's given by this equation. And it's a function of time, right? So the material basically has a residual strain characteristic to it that you're able to calculate. So this input vector space would look like this. And of course, I'm mapping that to just like the question that I just got about the fracture strain, or I'm mapping that to what the fracture strain would be, or some other damage variable that I'm calling D of X. That's a damage variable that's a function of my input vector space. So I'm able to predict that in totality. In sum, I can get the entire mechanical behavior of the structure once I have this neural network tuned based on the right input vector space quantities. So that's basically what I'm showing here in this example. Those are some of the input variables that I'm looking at from a mechanical standpoint.
I see. And so I kind of can see where you saw this kind of natural analogy to proteins, because you're starting, at least from this one, you're starting from some undeformed structure and it's getting some type of stress and getting deformed, just like a protein would denature and unfold. So that's, I see that's a very natural analogy. So kudos on even spotting that with what you were doing. It might not have been a lot of, mechanical engineers that could make that type of leak. So definitely kudos for that. Now, my second question, I wanted to ask you about when you're doing these things when you're doing these simulations on biological systems, are you taking into account solvation effects? And if so,
Yeah.
how are you doing that? Is
Yeah,
it still
so
like an all atom type of thing? Like
actually,
just
it's
putting a bunch of water
so
molecules?
we put them in water. I don't think I showed that here, but we solvate them. I do, you know, we start out in a vacuum, you know, so I start out with them in a vacuum and basically doing them, but I left out a lot of stuff in here, but we do solvation. So, and we can mimic the physiological characteristics of I think, let me see here. So this, yeah, but yeah, I'm doing, we put water, you know, put them in a water box or a water sphere or whatever like that. So we're taking solvation into consideration. I'm just not, but yeah. Yeah. So yes, we're taking solvation into consideration.
Okay, excellent. And I have one more question. So I know most of your concerns are like, you know, larger kind of macro scale mechanical stuff, but are you interested in possibly adding some quantum mechanics in there, like
get to
the
that level.
QM simulation?
Every time I try to get to that level, the NIH and the NSL run me off because they're like, you're trying to, I mean, I really want to get to that level. I've really got that on my mind, but I've even started some proposals and stuff. Yeah. So to answer your question, yes, I want to go back the other way.
Because what I'm thinking about, some of those parameters that you had where you're getting some of the numbers from experiment, some of that stuff you might be able to fit to quantum mechanical data.
I see.
And then that kind of opens up where, you know, whether an experiment has been done on the protein or not is irrelevant because you can, you know, you could just basically do the quantum mechanical simulations and get those parameters yourself.
We need to stay in touch, then. We definitely need to stay in touch. I need to get your information.
You guys gonna be talking like him and I were talking one day on a collaboration call.
Yeah,
and
we start going. It's over.
Like, it's just you playing.
Well,
tennis.
that was my last question, though.
Well, that's good. Hey, I appreciate that comment, but you got me thinking about something because I'm a
piggyback. Dr. Lawrence had to leave, but he sent his question to me to ask. And his question might build off of what Wallace is asking as well on the quantum level. Given that blood clotting is problematic in zero gravity environments, I'm wondering if his group has considered what, if any, contribution to the partition function comes from quantum gravity.
Sure, I have not, but that's a great question. That's a great question. I know what you're talking about. Yeah, we have not. But that's a good question. We need to stay in contact with that.
Okay. Well, I got you on the hypoxia stuff, so you know we can play with that in my lab. I got that. We can do a lot of stuff, and in real time, experimentally with fluorescing too. But I have even more ideas. I'm not going to hold.
That was a great question though about the quantum, because we need to stay in contact. I would like to hear more of your thoughts on that as well.
Definitely.
Okay.
I did just want to say one more thing just about your transparency about, you know, when you were switching fields, saying, you know, when you switch fields, you know, remember very little biology, kind of picking back off of what my colleague said. You know, we always tell students, you know, they can use this degree in chemistry as a springboard to anything that they want to do, regardless of whether you know a lot about it or not. It can be, you just use that education as a foundation and you can go learn and do anything. So, you know, I'm so glad that you were so transparent about when you switch fields, all that you had to learn on your own. I think that's crucial to what we try to tell the students here.
Yeah, because that's a good background for it, though. Chemistry,
yeah. We've had some conversations over the years just about that, with him and I just going at it and doing this. And so I really appreciate you, Dr. Everett, for like Wallace said, your transparency, letting the students know this is where I came from, right? And this is what the environment was. But look at me now, you know. I think that is imperative for them to see, you know, coming from humble beginnings, you know, and just being that work ethic. I still stay on that work ethic can get you, it can take you places you never thought you can go. So it may kill you along the way a few times. I've died a few times. I'm probably gonna die again before I get this proposal
I think we all have.
But no, I think this was
a lot of comments, a lot of negative feedback, a lot of like staying up late, like these guys talking to me.
I mean, he knows. See, I will text him probably or email him at like 12:30. My graduate student's on here, so he's gonna probably laugh at me at like 12:30 in the morning, and he'll say, "I see you up with me," and I was like, "Yeah." So we it's real, but you know, we are appreciative. I'm glad the timing was perfect, actually, even though we had to move it to the fall, but it was perfect for, I think, exactly what we all needed to hear. Any other questions?
I
No?
appreciate
Okay.
you all having me, and I appreciate
Yeah.
the feedback and the questions that you all had. You know, it's intrigued me and it has me thinking about some other stuff. And we should stay in contact. You can send out like maybe a list of people who attended. I think it would
Yeah,
be great
I got you.
to have that as well.
Yeah, I actually got something even better to add to that, but we'll talk about that later.
Okay.
It just opened up. It's I don't know. It's, the timing is impeccable because I had another conversation about something you brought up with a neighbor of mine, and it's just I think right now we need more than what we know, but just sharing
I'm
these
challenging my own
conversations and experiences.
institution on Black scientists and just involvement and basically
Yeah.
trying to change the whole structure and the narrative of everything involved. And, you know, I don't, you know, I'm getting to the point now, I'm like, I don't care